And we found this serendipitously, if you can use that term. There were a group of people who were working on preparing pigs, slaughtering them and such. This is an assembly line. The pigs would come in here, and they would be processed. Every place you see a rectangle is where there's a worker. The only people who developed the syndrome were the people who were moving the brains. And we learned, in fact, that what was going on was that there had been this introduction of this new method for collecting the pig brain, which was then sold in the Far East. You can see the eye of the pig, the sound of the pig, and so forth. And they were injecting air at very, very high pressure, which released myelin, which then resulted in antibodies directed against central nervous system, peripheral nervous system myelin, which then induced this disease. So it's important to have an open mind. Although I talk primarily about microbiology, it's important to realize that there are many triggers for the immune system. Toxins are key as well. And we are quite, I don't want to say, I don't know what the correct term would be, we are open-minded about thinking about mechanisms, because that's key, I think. We need to let the science drive the observation. Now, not many people talk about fungi in chronic disease, except in patient groups, and I'm sure some of you are concerned about problems with fungi. And to give you an example of recent work we published, where we've shown, in fact, that fungi are critical. So this is a disorder known as Kawasaki disease. It's been with us since the 1960s, and it causes a vasculitis and coronary arteritis, and at one point it was the most common cause of coronary heart disease in children in Japan, and it's still a major problem. And these children are typically treated with something called IVIG, which is an intravenous human serum immunoglobulin, which shuts off the immune response. And people have talked about a wide variety of infectious agents that's potentially linked to Kawasaki disease. None of them succeeded. And then there was a report a couple of years ago suggesting that there would be changes in wind patterns just prior to the onset of some sort of an outbreak of Kawasaki disease. It suggested that there might be something picked up over China that was then deposited in Japan. So we began looking at air that was sampled in specific regions. This then resulted in something which is absolutely transformational in, I think, in the discovery work that we need to do. When samples are present that have very, very low concentrations of the bacterium or the fungus of interest, you can be confounded simply because you don't have the ability to detect something. It's one thing to detect a needle in a haystack, but to detect a single needle in 10 haystacks is impossible. So the idea is, how do you clean things up? So we've been focusing a great deal in our pathogen discovery in ways in which we can help remove host material or other things that would confound our ability to detect these pathogens that are present at very low concentrations. And what we've learned is that the very tools that we use to do this work, whether it be the plastics or the enzymes or the buffers or any of the other solutions, are contaminated, not with something that will grow, but with something that will poison the downstream reactions. Methylbacteria is chiefly involved here. So we found ways in which we can eliminate this by using enzymes that cut these bacteria into very small pieces so that they don't amplify, and radiation what we can so that we can eliminate DNA. And this has allowed us to reduce, the amount of crap here, 73%, down to 4%, which gives you a bona fide, an accurate picture of what's actually present in the sample. And by taking this approach, we learned that in these troposphere filters that have been collected, that we found Candida as opposed to Aspergillus, which was present at the surface. This then resonated with some earlier work showing that Candida could cause an inflammation of the blood vessels in mice, and that then led to the discovery a little bit later, actually, probably about the same time our paper came out, with the idea that children who had this disorder had antibodies to Candida. Now, why is this important? But this then allows us, instead of just talking about ways in which we can treat this non-specifically, to actually begin to try to eliminate this trigger from these kids so that this is not a problem, and we can then begin to find out which one of these children is prone, why might they be prone as a result of genetic factors, or other factors that might trigger this. The other thing that we've been looking at, and I'm now beginning to talk now more about microbiome research, is to look in the environment, just as I said earlier, not only at animals, but also at animals that feed on animals. So these are phlebotomous insects, that are ticks. So we can look at mosquitoes, we can look at ticks, we can look at bedbugs and so forth. And the notion here is that some people who develop Lyme disease don't recover after antibiotic treatment. And I'm sure some of you will know people who fall into those categories. And the leading hypothesis has been that these people have some sort of persistent immunological illness as a result of this trigger, or that they may be feigning illness for secondary gain. I'm not saying I subscribe to any of these things. I'm just giving you the litany. And the alternative hypothesis that we wanted to entertain is that we might not be treating the appropriate agent. It's certainly something to consider. And the only way to do this, because no one has been able to culture anything from these patients that's not described there, has been to go back into the ticks, to ask what's in the ticks, then to make antibody tests, which we can go and use in the patients who have chronic Lyme disease, whatever that means, and see whether or not they're infected with viruses or bacteria that would not be treated appropriately with the antibiotics that are the classical regimen for treatment of Lyme disease. And we've discovered a number of different viruses that we're beginning to explore. And I have to be very frank with you in telling you that this is not a well-received hypothesis, although I think it's biologically plausible and it should be explored. And it's something that we expect will surprise many people when we're finally done. Several years ago, I was asked by the American Academy of Pediatrics and the U.S. Centers for Disease Control to try to assess what was happening with the reports of MMR vaccine and autism in the wake of Andrew Wakefield's work with the Lancet. We were unable to replicate his work, but we were impressed with the fact that children with autism have an increased frequency of gastrointestinal complaints. So we decided to ask how that might come about, and we began studying their gastrointestinal tissues, which led to the microbiome research, which I think is critical for thinking about CFSME. What we learned was that children with autism, not all, but several, had abnormalities in their abilities to process complex carbohydrates, disaccharides and polysaccharides, which have to be cleaved to monosaccharides, single sugars, in order to be transported across from the inside of the bowel through what's known as the enterocyte, and from there then into the blood. And there were dramatic reductions in the levels in the kids with autism spectrum disorders, listed here as ASP, for these various enzymes that break down these complex sugars, as well as the molecules that transport them from the inside of the bowel into the blood. That, in turn, resulted in changes in levels of specific bacteria that were present within the gastrointestinal tract. One in particular, Cetarel, which was very interesting, was apparently invasive because it resulted in antibody challenges as well, explaining why some of these kids responded to antibiotics, or restrictions in diet, or probiotics. And I think over the next several years, as we begin to understand more about the microflora of the gut, not just bacteria, but also fungi and viruses, you will find that there will be an increasing role to play with modulation of the intestinal microflora, which brings us to some of the work that we're now doing. Now, the other thing we learned was that there was another subset of kids within the same group who had decreased levels of other very important proteins, which I'm going to talk to you about now. One of them was called UGT17, and the others one of the three cytochrome enzymes. And these were important because these are the very enzymes that are critical for processing testosterone, nicotine, a whole range of things that are ever more common in the environment as we increasingly depend upon chemicals for preservation of foods, for flame retardants, for all kinds of things. So there are new vulnerabilities that are being uncovered that we didn't see 100 years ago. Whether this pertains to CFSME yet, I don't know, because we haven't done enough research in this area. This is one of the reasons why I think it's important that you focus on persuading your legislatures and your charities to invest accordingly. But my prediction is that you will find that there will be a subset that's purely genetic, a subset that will be toxic, a subset that will be microbial, and they will all act through certain final pathways to result in the disorder that you recognize in yourselves and in your loved ones.